structure. The Wilkinson Lectures celebrate the spirit of innovation, scientists who challenge conventions and open new ways of thinking about molecules. It is my great pleasure to introduce Professor Dame Coral Robinson, the Dr. Lees Professor of Chemistry at the University of Oxford, and also founding director of the Kavli Institute for Nanoscience Discovery, which explores life at the nanoscale through interdisciplinary research. Carol is best known for revolutionizing the use of mass spectrometry to study intact protein complexes, including membrane proteins, something once thought impossible. Her work overturned the long-held belief that these fragile assemblies could not survive in the gas phase. In other words, she proved that proteins can fly. How? Through pioneering adaptations of electrospray ionization in instrument conditions, she made it possible to preserve native-like structures. As someone fascinated by membrane proteins myself, I find it extraordinary how Carol's work opened entirely new ways to explore complex molecular machines. Clearly, Carol's work embodies Wilkinson's legacy, challenging assumptions, embracing new perspectives, and daring to see molecules in completely new ways. Carol's breakthroughs in mass spectrometry not only allow us to reveal the inner workings of proteins and even enable us to extract them from human tissues, including regions of the brain linked to psychiatric conditions. These insights not only transforming structural biology and the physical sciences, but are also paving the way for new medicines and approaches to mental health. Carol founded OMAS Technologies in 2016, which translates her discoveries into real-world applications. Her contributions, therefore, not only advance fundamental science and epitomize blue skies research, but also create opportunities for drug discovery and understanding diseases at the molecular level. I first met Carol as a very junior academic at Queen Mary University of London, and she immediately left a profound impression on me for 3 reasons. First, her unconventional career path. Becoming a technician aged 16, then heading straight into a PhD without an undergraduate degree, going on to become not only the first professor but in Cambridge, but then also in Oxford. This left me so intrigued that later, when I stumbled across a radio podcast and I remembered her name, I made the time to listen to it. What perhaps struck me most was her mentioning the fact that it's her technical expertise with the mass spectrometer, one may say virtuosity, that facilitates some of her breakthroughs in science. Second, I was impressed how elegantly dressed she was, at a time when it was somewhat fashionable to dress sloppily in academia. Lastly, this was at a time when I had to prove myself as a new lecturer on probation with a 6-month-old at home and many sleepless nights. I could simply not believe that Carol took 8 years out of academia to raise her 3 children. And here she is today, an eminent scientist, FRS, Dame, and, as I just learned, the awardee of 5 ERC grants, delivering this prestigious lecture. Before we begin, a quick housekeeping note. There's no planned fire alarm during this session. If one does sound, then please make your way outside to Perkins Green outside of this building. Now please join me in welcoming Professor Dame Carol Robinson for the Wilkinson Lecture.
Well, thank you very much. Wonderful introduction and a real pleasure to be here. I don't profess to have met Geoffrey Wilkinson, but I did study that book very carefully, as everybody did in my technical college. It was our sort of mantra for inorganic chemistry. Don't test me on it, but I did know it at the time. And I think it's probably still in print. I mean, it was an amazing book, and it deserves to be still in print and still used. So I hope you all have met it and used it as much as I did. So my research, as you've just heard eloquently introduced, is all about mass spectrometry, which is a very subtle way of measuring things very accurately. So it's a bit like a balance. And I've always thought life is a bit like a balance. It's very important to me to get the right balance, and so I'm often inspired by pictures of balances. I like balances. Somebody showed me some very elegant weighing today in the department here. I just thought, oh, that's so cool. Weighing, measuring, really me. I'm also intrigued by very early balances, and you may know this story of Antoine Lavoisier and his wife, Marianne. This is the first balance, and it was used for measuring the weight of tobacco. And maybe you know this story because later Unfortunately, he was accused of falsifying the weights of tobacco, which of course is a very important commodity, and was guillotined. And then she went on to marry Count Rumford, who discovered the third law of thermodynamics. So to me, that picture spells out that women's roles are often unrecognized in science. You have to wear this big dress, and she illustrated all of the diagrams in his papers and translated them from French into English. So she had a huge amount of power and influence back in those days. This was my hero. So when I was growing up in school, there wasn't so many women that we knew about in science, but Marie Curie was one of them, and I thought she was amazing. The way, this is her growing up. She's one of 5 children. She worked with her husband in this very dark, gloomy lab. She had 2 daughters, and amazing career. She won 2 Nobel Prizes, incredible. And one of her daughters then went on to win a different Nobel Prize, obviously. But I always remember, I was the president of the Royal Society of Chemistry, and we would talk to people about Marie Curie, and actually nobody had really heard about her. I hope you all have, but at that time, we'd go up to a bunch of 16-year-old girls, and I was trying to influence them to come into science, and say, "This is really great. Have you heard about Marie Curie?" And it's, Mariah Carey. Much more famous, much more of a role model. And then you realize, actually, Marie Curie, time for a change of image, perhaps. Maybe we need to get a bit more up to date. This is my start into chemistry. So this is my chemistry teacher. And I often tell the story that I'm not quite sure if it's an appropriate story, but I just, he's so good looking. I love chemistry. And I'm going to go, we're in an all-girls school. So any sort of young male teacher, we're just like, right, that's a really interesting subject. And one of the things I've often thought, if you work hard at something, you actually get quite good at it and you find it interesting. The previous teacher was a sort of, how would I describe her? Not great. We just thought, we don't want to be her, but we do like him. So we all started working incredibly well. The school record for GCSEs went right up when he came in. So I think there's something about inspirational teachers that really make a big difference. And for him, he was the person that I started working hard for. So I became the top of the chemistry class after showing no interest. So this is quite a big step. And then, of course, I made my career in chemistry. I was telling this story at a dinner in Oxford, and they said, well, you should write to him. And I thought, yeah, you're right. I really should say thank you. You probably didn't realize we all had a crush on you. We were all a bit inappropriate. But I wrote to him, and he said, oh, that's so nice. I'm 75 now. I'm in a chair, no longer the subject of schoolgirl crushes, but I do remember. And he said, I'll follow your career with much interest. So sadly, he passed away. But yeah, I still have big affection for, no longer a schoolgirl crush, but I still really like that time. And at that time, this is what labs looked like. This was a long time back. It was not the place you wanted to be. And I used to look at books of women in chemistry and things like that. There wasn't much to be going on. And this was a picture that kind of, I thought, oh, that's interesting. She's using a very early chromatogram, potentially mass spectrometer, not quite sure. She's got a very short skirt and a big sort of probe. It was very kind of, 70s look. I thought, I can do that. Short skirt, sit at a mass spectrometer and fiddle around with bits of equipment. But I just loved it from the first day I sat in front of that. That's my first mass spectrometer. I've since known, I'd say about 50 throughout my career, but that for me was a changing point. I went in, didn't know anything about it, just felt very inspired to be able to be given the chance to drive it and to operate it. And when I look back, I think, I actually think I was so young, I had no clue what I was doing. Why would they put me in charge of something like this? It's very expensive, one of the first in the pharmaceutical industry, in fact. But somehow managed to get it to work. we got a colour photographer at one stage, and this is me again. And this is actually showing, I quite like this one because it shows the actual flight path of the ions around the mass spectrometer. And we would separate them and work out whether or not someone had made the molecule that they intended to make. So this was my kind of journey from school to instrument, first instrument, and then to a mass spectrometry lab. And I haven't really left. I'm still there, having had a career break, but still fascinated by what I see in the mass spec. What is a mass spec? I thought I'd put this in case I had some, you all look like you know what a mass spec is. But in case you didn't, it's that thing that you would see at the airport when you go through security. It's doing a swab sometimes of the things in your bag, or it's taking some air from around you, and it's measuring volatile molecules or things that it's rubbed off your luggage to make sure you're not a drug or explosive carrier or something like that. And very quick readout. So it's a very robust thing. Very small molecules, very volatile things, but it can pick up cocaine, explosives very readily and very accurately. But now think much bigger. So where I want to take it, and where we've been taking it, is into proteins, which are very much larger, and protein complexes, and to see what we can learn about them, because you all know, I'm sure, proteomics is a hugely important and very well documented field. It's chopped up proteins, and it identifies and builds a picture. I want to take top down, so go from the very big complex, and then see if I can maintain it, and then see what we can learn about its interactions, etc. This is my PhD. Another great and important person in my life was Dudley Williams, who, imagine that you're a professor at Cambridge, and then someone comes along and they haven't got a proper degree and they've got a lot of experience, but you're not really sure if they're going to be able to make it or not. And he gave me a lot of confidence and gave me a chance. So I was very fortunate to walk into his office. And what he always told me was, what he always said to me was, why are we doing this? Is it because we can or because it's interesting? And I've always held that, why are we doing these experiments? Are they actually going to change? Is the man or the woman in the street going to care? Why do we care? So I think that's a very important thing to always have at the back of your mind, and that's what I learned during my PhD, because quite often I'd get quite techie. Look what I can do, and then, actually we don't really care that you can do that. It's not really interesting because you can't solve a problem. So very important. And during my career break, very important time for mass spectrometry. It went from the sort of small molecules that I was used to studying into a new world of proteins and very large proteins and oligonucleotides, not complexes at this stage, but individual proteins and identifying proteins. Incredibly important work by these people here. Actually, I like this because I think it's a really nice analogy. So John Fenn, brilliant scientist, people would say to him, so what have you actually done? And he said, I gave elephants wings, because till that time, only very small molecules could fly. But once you could fly a protein, you could start to fly potentially a whole machine. So I like that analogy very much. This is my return from my career break. You may know these people, you may not, it doesn't really matter, but I went to Oxford as a technician, basically, again, back at the start, as a postdoc, but a research services kind of support person, which was fine. I was collaborating, and I was thinking, who's going to be my mentor? I had two bosses, on the left, Jack Baldwin, and on the right, Chris Dobson. Who was I going to work for? Who was going to be the person that kind of changed my career? And I learned very early on that somebody, I don't know if I've got a pointer, maybe not, but somebody who puts "and in part" as an author is actually not really respecting you. So you're a half an author on a paper, which actually I did all the work for this because it was an O18 exchange on an enzyme, measured very, very carefully. But you've probably never seen part authors, but it's a thing that I thought was totally demoralizing, because I thought, well, I actually worked harder than the rest of the people, but still I'm a part person on this paper. So I learned, choose your collaborators carefully, and they have to be the people that really respect what you're bringing to the table. And if they think, that's just like a melting point, it's not that important, I'm not going to give you full credit, then that's actually not really somebody you want to work with. You want to work with people who really get behind what you're doing and really support your work. So I moved very quickly to work with Chris Dobson. We were interested in how proteins fold from unfolded denatured states back into compact native states. I'm sure you know the problem. It's been talked about a lot. With electrospray, we were looking at how these molecules, we could get them to fly, but we didn't want them to unfold. So as I mentioned, traditionally it was unfolding, it was chopping, it was proteomics. What if we didn't unfold them? What if we kept them together? What would we learn about their interactions? And this is a schematic, and I see it like this. You have something that's happy in water, you want to give it wings, you want to take it through the gas phase, through the atmosphere, and then measure it and make sure it's kept on its wings and all of its bits, because you really want to learn as much as you can about it. And these became my friends, mentors, and collaborators during these protein folding days. Andrew Maranka at Yale, Sheena Radford at Leeds, and Chris Dobson, sadly no longer here, but was in Cambridge and really helped my career. And he really appreciated what I did. He always said, I had magic fingers with the mass spec. It was a pleasure to work with him, because he encouraged me. Every time I said, I'm not really ready to apply for something, he said, no, you are, go now. And I think that's such an important message for people that I mentor as well now. But lots of people said, well, folding in a test tube, what's that about? Doesn't really mean anything. Folding takes place in the cell, not in the cuvette that you're measuring it in with your hydrogen exchange. All very good, but it's not really relevant to the world. You need chaperones, you need ribosomes, you need to recreate the cellular environment. So our next stop was to make it more cellular, introduce the molecular chaperones, and this was my sort of favorite experiment for a long time. Form the molecular chaperone complex, label it, hydrogen deuterium exchange, get a change in mass, break it up, and then have a look at it. Super proud of that, but this unfortunately got changed. However, I'm going from Mac to PC. But there was a lot of commentaries that were very against this work at the time, and this is a sort of another lesson in life, is that sometimes people are going to try and take your work down and write bad stuff about it. If you believe it and you know that it's right, you can still keep going. And I think for me, this commentary that was published by very revered scientists in PNAS was a bit of a turning point, because people would say, whoa, I would submit another paper and they said, well, did you not read the review that was written about this work in PNAS? And I'd say, well, of course I read it. I don't agree with it. This is my answer. But for a long time, there was quite a resistance to what I was doing. And then I remember people saying to me, just do proteomics. And I said, no, I don't want to do that. I really want to do this because I believe in it. And I think if you believe in something, then you have every right to keep going. And one of the turning points was to show that this GROEL thing, which is 14 subunits, could actually stay together. That's a very poor spectrum by today's standards, much better now. So this is at 800,000 molecular weight. And if you think, that's quite a big thing to trundle through a mass spectrometer when most things would be coming out below a few hundred at that stage. So this was a big kind of turning point, whereby we knew that these complexes would stay together and have the right stoichiometry. We didn't know much about the shape or whether they partly unfolded or any of those things, but we knew that there was a 14-mer and a 7-mer, so the topology was, of course, right. And then fast forward another decade or so, and we became very interested in seeing if we could do the same things for membrane proteins. And this was this slide has lots of memories for me. One was, this is very old now, so excuse the artwork, but my son drew this with me, which was a very nice activity because he didn't think anything I ever did was particularly interesting. So, okay, I'm going to tell you about this. It's going to be great. We're going to try and get the front cover of Science. It's going to be your artwork will be on the front. And of course it wasn't, because that's a very simple image. It did make the inside cover. And so I'm super proud of that piece of, you know, you're going to sit, you're going to learn what I do all day, and you are going to be interested. It was the first time I'd managed to take a membrane protein complex in a sort of detergent bubble and put it into the mass spectrometer without it falling apart, breaking the mass spec, sticking it up with too much detergent, etc. So that was a big day for me. Well, it took several days. And I also remember that we sent it to Science and the editor said, well, yes, okay, but you need to think a bit more about the title, which was that title. And I just said, I've spent 3 months thinking about the title. You think I just made it up? Anyway, so they didn't approve of that title, but that title comes back at the end of my talk, and I'll show you that actually turned out to be correct, because we didn't really know that it was the micelle that was protecting. We assumed that it was because the micelle was there. And all the theory would have said that they would fall apart because it's there's a vacuum, there's no hydrophobic effect anymore. How's it going to survive? So we decided that it was the micelle. Fast forward again until when Dror was in the lab with me, and we were very interested in trying to capture receptor signaling. So imagine this, you're looking at me and your eye, well, you might not be, but that's okay. Your eye is reading a message, and this is translated through rhodopsin down through your signaling pathway. So rhodopsin is a GPCR in the retina of your eye, and it's signaling the image that you see on the screen here. So what if we could take the retina from a cow eye, reconstruct that signaling pathway in the gas phase of the mass spectrometer? It was a kind of crazily mad experiment that you needed a dark room because all of the eyes had to be set to a start point, which is dark adapted. Then you shine on the light and they start signaling. Could we capture that? So let's move a mass spec into a dark room, or make a dark room around a mass spec. This is a lockdown project because not many people wanted to work in the dark, surprisingly. Here's a red lit room. Here's my mass spectrometer. Here is the spray coming into the mass spectrometer. Here's the dark, and here's my masked up student Sion. And if you look here, you can see peaks in the mass spectrum, and she'll show you close up. And then what she does is she switches on the white light. The rhodopsin then wakes up, starts signaling. And if you're really into mass spec, you can start to monitor the mass change. The two peaks change as retinol is lost from rhodopsin to opsin, and you can measure that in real time. Now, I thought that was just so fascinating to be able to recreate a sort of visual cycle in a mass spec. But you can imagine not everybody thought it was as fascinating as we did. But what we did find was that actually inside the membrane, in situ, if you like, it was actually a slower reaction than when we reconstitute it in a micelle. And we argue that that's because of the force of the membrane, which kind of stops the flopping around and the signaling quite so rapidly of the rhodopsin. This is, oh, sorry, it went too fast there. This is not a pointer. So this is Sion and Dror who did these experiments. And on the left is the dark, at the top, dark rhodopsin and opsin. And you see as the ratio changes because the retinol comes out, you excite and it comes out. But what actually I found fascinating was that Sion maybe got a little bit bored in the red lit room. So she started to flash on the light. It starts its decay, and you watch it decay, and that's the curve that we see in the middle. But then if you click back on the white light, it kicked up again. And at first we thought, well, what is that? Because that's a very fast reaction. What's going on? It can't be enzymatic. It's way too quick. And then we realized that it was actually, what happens is that the retinol goes back in again. So because it's in the membrane, it's held very close to the rhodopsin, and a kick of light can push it back in and they change its conformation. So if you look up in the left there, we have the retinol slightly outside. We hit it with the light and it comes back in. I'm sorry I can't point to it. It's the pink bit up in the top corner there where the cis retinol is coming back in again to form opsin, which then goes through the cycle again. So we found this regeneration mechanism because it was still in the membrane. And, although there's 50 years of research on rhodopsin, you might think, why even bother? Almost all of it was in detergent or membrane mimetics. So actually going from the native membrane was very exciting for us. And then we could see right down to PDE6, which is at the end of this reaction cycle. Beautifully artistically shown. The red, this is a photo of the mass spec. Obviously not a photo of the membrane, but coming down the light into the GPCR, down that signaling pathway to PDE6. We realized then that we had also a great way of doing drug discovery. I'm always interested in drug discovery. Given my start at Pfizer, we could recreate this kind of signaling pathway and add drugs and see how they changed the signaling pathway in a much more sort of in situ style arrangement. And this showed the importance of prenylated proteins, which is that they form this kind of hydrophobic tunnel whereby the drugs can bind across different targets. Fast forward. What are you going to do? You've looked at membrane signaling in the eye. We've done lots of other things that I haven't talked about, got quite good at membrane proteins more generally. But perhaps partly inspired by the pandemic, I noticed the very different effects of the pandemic on students and just people generally. Some became quite resilient, some became more depressed, some were fine, others were not. Why was that? We started to think, what if we could look inside the brain and get a sort of molecular autopsy of why somebody decided to take their life. Now, that was a big, dark question, and how were we going to do this? So we obviously had to collaborate with a lot of people who knew a lot more about the brain than I did. I wrote a proposal, and somebody said, great, but you know nothing about the brain. I said, yep, I know. I'll work with people who do. And so people told me which regions to look at, which ones were associated with depression. We then contacted brain banks and said, could we have age and sex match control for suicide? And then we're going to compare them. We don't know what we're going to see, but we're going to see if we can pull out GPCRs and receptors and transporters that might be involved in a depressive phenotype. And these were the people who, some of them thought it was a bit of a macabre project, but I just say, well, if we find something, it'll be amazing, and we can then use that to help understand. And this, I'm sure you probably know it, and some of you probably know it better than me. It's across the synapse. It's the glutamate receptor signaling, and it gets packaged by glutamine synthetase, which changes it forms glutamate, and then it's transmitted across the synapse via vesicular glutamate transporter, and then is received by metabotropic glutamate receptors, which takes too long to say. So I'm just going to call them mGLURs, and they are thought to be responsible for mood. So maybe there might be some differences there, was the theory. And this is the way we think about it. We think, okay, we're going to go into these various regions of the brain. We're going to hook out these receptors. We could either have them in vesicles, GPCRs and solute carriers. They're going to be multi-component complexes. And we'd like to learn about the lipids and the ligands that are associated with them. Because it may be that it's just a very simple chemical change of different lipids or ligands that are associated, or it may be that they're completely different, different isoforms or different proteins, or they may just be configured differently. And we're going to look at the post-translational modifications. They may be got hyperphosphorylated, for example, in overactive brain, or all sorts of questions. Bit of technology development, which is also one of my favorite things. Here comes my vesicle through the mass spectrometer. I'm going to hold it in the linear ion trap, and I'm going to fire a laser, an infrared laser at it, and I'm going to break it. And I'm going to see what all the components are in the Orbitrap. And get an intact mass spectrum, or hold them there longer, hit them harder. Hopefully this works. It's a bit like using a mass spec. And now all the fragments. And from the fragments, which are appearing up there, we should be able to read off the sequence, read off any modifications, but also required a lot of software development. So this is not going to be telling you about proteins that are already in the PDB. It's going to be telling you about proteins that maybe haven't been seen before. Discovery mode, we call it, for obvious reasons. We need to learn how they break and how GPCRs break, which is also quite interesting. Then we did some further optimization, which is probably not of super interest to this audience. But then we came to this picture, which is what are the proteins that are in the regions of interest and how abundant are they? And we could see that myelin, for example, is the blue one at the top there. And then we come down to the two M-glurs that we're interested in, the pink, the purple and the green, and they're not visible to native MS. So that's a bit of a blow. Can't see them in their native environment. So we're going to have to think of a way of enriching them. So we're going to use nanobodies, which you may have heard of. These are these kind of super soluble antibodies that you can stick on a protein and give it a handle, pull it out of a tissue, for example, a brain, but in fact any tissue that you're interested in now. So it's an incredibly powerful tool. And we did this with the two that we thought were most likely to be different in depression, the vesicular glutamate and the metabotropic. This is from a single mouse brain, so I didn't know if this was ever going to work. Would we ever get enough material? Fortunately, we did, and we could see a monomer and a dimer for the mGlu2, and the vGlu1 was all there. But then came the question, how do you make a depressed mouse phenotype? Which, of course, you can't. So you mimic it with a chronic social feat distress experiment, which is kind of similar to depression. And I didn't do these experiments, so please don't judge. But we collaborated with somebody at Mount Sinai who has this model whereby you have a more aggressive breeder mouse come in and sort of beat up the mice for a bit. And then eventually, some of them become susceptible to this bully coming in. And they're called the susceptible group, and then the control group never see the bully. So that's kind of the lucky group. And then we measure the brain from them, and we measure their social interaction score. So you then take them out and introduce them to a mouse they've never met before. And most mice are quite curious. I used to keep mice, so I find this quite hard. Most mice are quite curious, and so a new mouse, they go over and suss it out, as we say. Whereas the bully mouse, the defeated mouse, introduce it to a new mouse, and it just hides. So that's what we're measuring. The sus mouse has a much lower social interaction ratio, so it doesn't want to meet the other mouse, whereas the control mice are quite happy to go over and say hi, whatever they do. So we then measured the control and the sus mouse, and this was the best bit because we found a difference. So if you can look at those two spectra, I know probably a bit boring looking at spectra. I love them still. However, if you look at the grey bit to the left-hand side, you'll see that the peaks there are smaller. And that means on the bottom, on the control, smaller than in the depressed group. And that means that there's more monomer in the sus group. And we know that this dimer has to be a dimer to be active. So if you've got any monomerization, it can't be active because you need 2 halves for the active working receptor. So this was our first clue. That's interesting. There's something really statistically significantly different. And then we tried it on the human brain, and again, this was from Edinburgh Brain Bank, very grateful to them. So the control person who died from other causes has metabotropic glutamate, which is a very good dimer, and another protein I won't talk about at the moment unless you're interested. And then the depressed person has a much higher ratio of the falling apart of this receptor. So we could, really exciting. It was significantly different, of course. And then we think, well, why? Why is that falling apart? Maybe it's to do with the lipids that are interdigitated. So you've got this dimer, and for some reason it's able to come apart in the depressed or the sus mouse, but it doesn't in the control. So what keeps it together? Given my interest in lipids, I think there's something in there that's holding them together. It's likely a lipid. Let's do the lipidomics around the receptors. And we found that in both mice and human, the control in pink has a lot of phosphatidylserine, but the depressed mouse or person has much less. So this was our first clue that perhaps there's a link between your lipidome and your proteome that is conferring this phenotype. And you can see that on the graph there, there's a strong correlation with the amount of PS and the amount of dimer that you have. So we were super excited, went to look at the interface, looked at it with a structural biologist. He was more than happy to model in phosphatidylserine. And then we did some spike recovery experiments. We take the metabotropic glutamate receptor that we made, we dilute it, it falls apart to the monomer, we add back in the lipid and it recovers itself. So clearly, I say clearly, a lipid-mediated dimerization. So this is like the staple that holds the 2 halves together. This is kind of something I spent too many hours studying, I would say, but I enjoyed every moment of it. The 2 at the bottom are controls, and the 2 at the top are sadly suicide brain. And you can see that there's a shift, a right shift. And that's because, well, I won't spoil the story yet. And there's also an increase in the monomer. And then I looked, as I said, I have 20 or so of these, and I studied them for days and weeks. Probably shouldn't have, but I just loved it. Because if I look at the patient 2, the control 2, he was veering on the side of my depressed group. And I was like, oh, this isn't really working, because I reckon that I could look at a spectrum and say depressed or control. But that person bothered me because he looked to be in the depressed group, but he was a control. And then we did what we should have done from the start, which was to buy the notes from the doctor. And he said, well, actually, he had major depressive disorder, and he was on 4 antidepressants, and he had a heart attack, coronary artery, but that was a comorbidity. He was actually majorly depressed and had treatment-resistant depression. So the moral of the story is buy the notes from the start, and then I wouldn't have had him in the wrong group. And also think about the grouping, because if you ask for a suicide group, then you're assuming that that's a black and white thing, and the other group is not. But what about all those people in the middle who sadly didn't die by suicide, but may have had this major depressive disorder for a long time of their life, and so their brain receptors could have changed into this pattern. So physical scientists can't resist setting up KD measurements and trying to work out what things should be there. We worked out the mole fraction of the M-GLUR in the brain, and we came up with this theory that it was the loss of PS that drove the concentration of the monomer, which then led to a heterodimerization. So there's a heterodimer forming in the brain with mGlu3. And I'm not going to go through all of that because I realize it's getting late and this is getting a bit complicated. So we have these heterodimers forming by a rogue mGlu2 monomer. And this is the theory that we have. And then the purple one actually has a higher affinity for glutamate. So it starts to pick up glutamate more, and then it does endocytosis. But the left-hand one, the green one, actually is a better signaler. So you've got this kind of now differently tuned, attenuated response, which is provoked by loss of PS. Of course, no one's going to believe something that you've discovered in the mass spec, so you better get and do some other experiments, because you can't just go with what we considered a slam dunk with mass spec. You better do some live cell imaging. Had to upskill ourselves to do this, and we found that you could, of course, create an increase in phosphatidylserine in the membranes of these living cells or in the mouse head. So we did a gene transfer into the mouse, and we did a gene transfer into the live cells, and I can't point, but on the upper right there we have green. This is increased PS, leading to greater localization in the membrane of mGlu2. So it's a complementation assay. You've got 1 to 10 and GFP on the 11. So when they come together, they fluoresce. They come together better in the PS environment. And this is my favorite, one of my favorite experiments, because if you have your control mouse and your CCSD, or your sus mouse, the one who's susceptible to bullying, and then you give an AAV with GFP on it, which shouldn't really do anything, the sus mouse is still sussed, he's still socially defeated. But if you do the same experiment, but give the gene which synthesizes more phosphatidylserine, the sus mouse is now resilient mouse. He has a better chance of surviving the bullying that went on. So for me, this was amazing that we could do this, and helped validate our mass spec. We then did transcriptomics and saw that actually it's not just about depression, it's about stress. Chronic stress early in life is a major prognostic indicator for later life depression. So we thought, oh, stress is actually easy to recreate chemically. We can do this. So we did lots of stress experiments with live cell imaging, and showed again that stress actually increased the ability of the homodimer to come away and form the heterodimer. And that's shown in the red after we've induced this stress, and we can show an increase there. So this is our molecular adaptations to stress, which is that actually the homodimers separate and then mix up and form other heterodimers, which changes your signaling pathway. And there's lots of different reasons for that, which I won't go into. I want to give you one more mass spec. Let me just see how I'm doing. Right, quick mass spec thing, and then you'll be free for the drinks. So this is a great collaboration with Stefan Raschenberg in my group. So you remember I told you at the start that we believed that my cells protected our membrane proteins from solution to gas phase. And this is a bold title. And now we have a way of looking at this. And you will know, of course, that EM is amazing. Almost as good as mass spec, but, and we could now imagine an experiment where we put something into the mass spec, and then we pull it out, and then we look at it in the EM. So if I'm right, this isn't the only reason we do it, to prove if you're right or wrong, but just because you're interested. If we can put something into the mass spec from, sorry, that didn't work. I don't know what's going on there. Did you see something happen? Right. So here comes the beam round through the Orbitrap. That's what I've measured. That's my mass spectrum. And now I'm going to give it a different route and take it along to my cryogenic chamber and pull it out. And this is now ready for EM. And I'm going to either ice coat it in that final cryogenic chamber, or I'm just going to take it downstairs. It's great to have it all in the same building. And I'm going to look to see what is landed on my grid. I'm sorry, there's some movement in all of the text there. And this is 2 AMTB dropping from the mass spec onto the grid. And you can see at the top there, there's lots and lots of peaks, because actually having detergent, DDM, individual molecules of detergent stuck to your protein actually helps protect it. And it's better structure in the top than it is in the bottom. So if you get a beautiful mass spectrum in which you've got rid of all your detergent, then you get less resolution in your structure. Lots of stuff, lots of our favorite complexes, not yet from brain, that's the next step, but taking AMTB, for example, on PEF, and they're all reasonable in my book. They all have below 3 angstroms resolution, well, especially AMTB, the others, no, not really, not yet. And we vary the ice, we vary the deposition, we vary the activation. But AMTB was very interesting because we got a very good structure. We also did, oh, I forgot to show you. Yeah, we also did the plunge frozen counterpart, so you can land on the grid, run downstairs with your gas phase, or you can put it on a plunge frozen grid as the normal workflow is. So the bottom row is the plunge frozen control. And the thing to note there is, in all the plunge frozen row, there's a little halo of detergent micelle, because that's how you do it. You put it in detergent, you pop it on the grid, and you plunge freeze it and run to the microscope. Top row, no detergent. Interesting, because of course it goes in with detergent, but the passage through the mass spectrometer, you're shaking it off. So what effect does that have? Now we're going to compare the plunge frozen in pink with the electrospray deposition, and you can see very, very close, we like to think. We look at all the bits, and actually my postdoc student, she said, well, there's a bit of a difference. I don't really like the fact that it's different. And I said, well, what's the difference? Let's have a look. And it turned out that there was an extra 15 residues at the C-terminus that could be observed in the electrospray one. So you think, well, why is that piece actually really well preserved in the gas phase, but not in solution? And then when you look at it, it's all about electrostatic interactions that are well maintained in vacuum and less well maintained in solution. And in fact, if you look at this complex that was published by another group, you can see that actually that green region that we see in the electrospray method is actually intact in this region in the interaction with K, whatever that is. So, yeah, differences, sometimes interesting, sometimes there's more movement in the loops, for example, but then sometimes you may learn extra structure. It's not taken off yet as a means to surpass cryo-EM, but it's an interesting adjunct, because if you imagine, you could separate via mass, and this, I think, leads to a lot of exciting opportunities in separating, for example, ligand-bound, ligand-free, etc. So with that, I go back to my cells do protect. Remember this title that was so controversial with the science editor who said, think about it some more. And I was like, well, actually, it's true. This was my picture from Science. My complex in a bubble came out of the bubble a bit, and you can see some ugly peaks. But actually, when you look at the structure, this is now some 17 years later, you can see that actually the structure was this. It's a lesson in perseverance, really, isn't it? 17 years to get to there. So what do we see? Some things I didn't talk about, rotary ATPases, very nice work from mitochondrial membranes, but across the eye, into the membrane, looking at complexes on EM grids, and now regions of the brain. But regions of the brain is just one thing. I now feel I could sort of go off to all sorts of organs with organs of choice with nanobodies and try and pull out complexes actually from tissue. And from tissue, is I think where we need to be, because that's where you're going to find things. Huge numbers of people, tooth bank, wonderful groups over the years, supporters and from my company a bit, and from Kavli, from manufacturers actually shouldn't underplay the part that they made in helping us design new equipment and getting it all to work. And of course, thank you for your attention. And this is my proud slide of all the people all around the world who I've worked with, and they've gone off and started their group. I'm always, people say to me, yes, but I'm working in industry, I'm not on the slide. But I always say that's confidential. This is out there in the public domain, and I'm super proud of them all in their academic roles. So thank you for your attention. I'm happy to take any questions.
Thank you so much for this inspirational talk to the fourth. Before we thank you and go for drinks, we have time for a few questions. So I think it would be great to get some from the audience. I guess, okay.
Thanks. It's on?
Yeah.
Yeah, okay. Yeah, excellent talk, of course. What else do you think that PS is doing? So you focus on that one receptor, but do you have any clues about any others?
Yes, I do. It's actually, of course, much more complicated than I thought. We've also found a lot of plasminal ether-type lipids, which I think have a response to reactive oxygen and stress. So that's taken us off in a completely new area now, because they are very up in abundance in depressed brains. So are they there as a kind of way of protecting the receptors, potentially, because they absorb these reactive oxygen species? Or you maybe know, you're the expert. I just find it very interesting that there's a lot more to it than we thought. We thought, oh, it's just one lipid, but actually it's not. And there's hexaceramides as well, which are also maybe playing a role. Lots more work to do there, I think.
Maybe I'll ask a question while people getting the things. So it's incredible to me that you're able to go to the brain and get all these signals out. And I think you hinted at the heterogeneity and why. Can you tell us a bit more technical perhaps, how you, because this is really, after all, what we're after. Right?
There's a lot of, so what is it you want to see is always the question. Do you want to see lipids, metabolites, kind of heterogeneity and mess? In another word, I can't think of a better word, but chemical heterogeneity equals bad peaks. You will have noticed peaks are very bad. Not textbook, but that's because we've kept all the sugars on, because we also reasoned and we wasted or spent a huge amount of time defining all the sugars, thinking could be in the sugars, could be real difference in the glycans that are modulating the interactions in the receptors. I'd say a year spent on cataloguing all of that, no difference. But yes, you do want to keep the heterogeneity, because that could be, if you just get the bare sequence, we didn't find anything in the sequencing. Mm-hmm. The sequences were the same. The abundances were more or less the same, give or take a little bit of one of the receptors was a bit different. But basically, it's all in, I think, all in the arrangement and the interactions with the lipids. And it's been known for quite some time, I think, at least I think it's known, that there's a correlation between the lipidome in depression and healthy individuals, which may be, well, lots of reasons. One of them is diet related and all of that. It's very much more as somebody, when I was starting this project, somebody said to me, it's never gonna work. It was a Nobel laureate came into my office, not to name drop, but he said, I don't know what you're doing that for, because you're never going to get to the bottom, because, maybe you take alcohol, maybe you do this. How are you ever going to pass all that out? And of course you can't. But if you can form two groups and there's a definite difference, then maybe you have a start.
Yeah. Oscar.
So that kind of feeds into my question. How reversible is it? If it is diet or so forth, can you actually, in the absence of drug intervention, can you see in patients the markers go back to what they would with diet changing?
I like to think, so the question maybe you didn't hear was, what if you change that diet? Could you do a sort of remedy almost? And I like this because one of the things we had to do, and I think it was a good thing, was talk to a patient group. And it was so informative to me because I was, I found this, I'm so excited, I'm going to find you a drug. And they were just like, no, we don't want any more drugs, done enough drugs, not interested. Oh, what if I gave you a nutraceutical, which was my nutritional pill that would kind of go, oh yeah, that would be fine. So I'm thinking, along your lines that I could give you a little PS pill and maybe it would actually work. And that's why we did it. I just said, let's just feed the mice PS. And there was all this stuff about, will it get across the blood-brain barrier? Will it even get there? Which is why we did the injection in the head. But I reckon that we could also do the feeding and maybe enough of it will get in. And I also like this, I was in my office thinking about how I'd make this nutraceutical. And of course, Chinese traditional medicine is years ahead of me. They already have it. So they already sell PS for major depressive disorder. And I thought, that's amazing. I know how that works now. It's sorting out your receptors in your head. But they make it from some kind of fermented sort of bean type thing. Doesn't look super appealing, but obviously it's really good for you. So I've always found it fascinating how they knew all these things thousands of years before they had a mass spectrometer or anything like that. So, yeah, amazing.
Last question from Nazida.
Thank you for a great talk. I just had a very quick question with regards to whether you can apply your tools and mass spec techniques to studying the dark proteome at all.
What do you consider the dark proteome?
So structures of proteins that we don't know much about their folding or their 3D overall structure.
I'm sure we can, but I haven't done that, I have to say. You would have to work out how you were going to find them in the dark, I guess, is the first question. A lot of people talk about the dark proteome to me, and I never quite know where it is or how to get at it, but I think it's a very interesting question. Do you look at the dark proteome? Yes, yeah.
Well, please join me in thanking our speaker again. And I think on top of the amazing science, I think it's also inspirational for us to, and I think for our younger people here to hear that you do have to persist in what you believe in, and you have to go for it and not give up and ask the good questions together with the technological advance. And never forget that there are people who inspire us from young age, people who open doors for us and who sponsor us. And I think, thank you for all of this and for being here.
My pleasure.
Hi, thanks. Can everyone hear me okay? Brilliant. So, for those of you who don't know me, my name's Oscar Says. I'm the head of the Department of Chemistry. I just wanted to say a few words before setting us outside for the drinks reception. So the first thing is to, thank you all very much for coming today. There's a multitude of people here from the department, from local schools, from companies that we kind of work with, from our community. Thank you very, very much for coming. It's a very warm welcome. We'd like to wish you here to this evening's Sir Geoffrey Wilkinson lecture. The first person I want to thank, and above all, is obviously Professor Dame Carol Robinson. I met Carol about 14 years ago, and I was a lipid person at the time, and she gave a lecture which kind of changed my life, because at the time understanding how membranes correlated with protein function was kind of an emerging field. And like she said, people often said it was too complex a thing to understand. You've got 2 million proteins, you've got 25,000 types of lipids. Every single cell's making these in abundance. How do you kind of, extract engineering rules from that? And she was the first person who showed me that it would be possible. And I think she's kind of pushed the boundary ever since then in a way that kind of no one else has in the world. So I just, I want to say that from a personal level, I find it absolutely kind of inspiration. And I think tonight's talk summarised that. It was illuminating, it was inspirational. It shows you someone who did not accept that things couldn't be done. Someone who basically challenged kind of dogma all the time. And as you saw before, when people put barriers in front of her, she kind of just walked over them and kind of carried on forwards. And I think, just wanted to thank a big warm welcome for having her today. And if you could join me in thanking her, that'd be much appreciated. Thank you. So alongside Cal, there are a couple of other thank yous I wanted to make. So the first one is to, where's Cider? Cider, you around? She's hidden herself, so she's the person who actually organised tonight. So yeah, she's outside. So I'm not going to make her come in and do some sort of surprise thing. We sort of clap for her, but she organises pretty much all on her own. So normally in the past we had 4 or 5 people running this, but she did the invitation list, reaching out to people, logistics, the Eventbrite, everything. So, in a second, if you just come in, let's give a warm hand of applause. But she has been, with the support of actually Sarah and Mark, done an absolutely fantastic job. And that includes kind of, you won't have seen, but as well as the lecture that Carol gave today, she's been kind of really, really kind with her time, meeting academics, meeting people from local schools, meeting kind of PhD students, inspiring us, and decided to help kind of put that all together. I also want to thank our host of the day, so the two Nicks, Nick Long and Nick Harrison, who met earlier on today with Carol when she first arrived. But above all, I want to thank Maxi and Sophia for anchoring the day and making it such a wonderful event. Maxi for a wonderful introduction, and Sophia for sort of guiding us through a very thoughtful and engaging Q&A. And then finally, you folks, the audience, both things. As I said, I started that, I want to finish with that. For being in person, many people are online. We had something like 350, 400 people apply to attend today's lecture, both in person and online. So I wanted to basically ask you to bring your hands together to thank the people who organised this. Carol again, yourselves for attending, and please welcome me. Thank you. And last welcome is to the AV team. And Panji, who's been walking around really subtly in the background, taking photographs. I want to give them a big round of applause as well. Thank you. And thanks with that, one thing I will do when I get home is open up my copy of that book. It was actually when I got chemistry prize at school, it was the first textbook I ever bought. I think I've got the 3rd edition, which is actually the one up on the screen, which has made me feel very old suddenly. But in Sir Geoffrey's kind of spirit, let's carry on forward, let's continue to explore, let's continue to kind of break boundaries, but above all, let's also continue to kind of support each other. You saw in today's lecture that the spirit, that last slide that Carol showed, the kind of collaborative network, it's how I think of science, kind of not in isolation, kind of working with people. So folks, with that, thank you again very much for coming. There's some nibbles outside, there's some wine, I think there's some milk, there's some orange juice, there's kind of something for everybody. So if you join me coming down here, let's go outside and enjoy the rest of the evening. Thanks a lot, folks. Cheers.